some big discoveries, yeah? Some big discoveries, anything new, yeah? Lady, yeah? Anybody hands up? No? This is a very boring week, yeah? No? We're just waiting a few more minutes, yeah? Nézzetem a kapcsolatot, mert már befeléket a rendszerben. Oh, it's a good plan. That's a good plan. But you need to grow first. Yeah. I'm also try not to do too much about this. You don't know. Once you need what you should see what it no idea.
Sorry. Yep, I'm ready to help you. Yep. This is a save. Okay, this is a wireless microphone. Let me put this on, okay? Yep. This is into your pocket. Right. And that's right. And that's
Great.
okay. Thank you.
Thanks for your help. Yeah.
Okay. Yeah. I'd like to introduce Dr. Nick Christ, one of my excellent colleagues, and I asked Nick what should I tell about him, and he said, I'm a lovely guy, and indeed, he's a lovely guy. So I'm very glad that he adventured to come over this part of the world, and hopefully he will visit us again. He just reminded me that Nick did a PhD at University of Virginia, and then he was a postdoc in Indiana, and since 2008, right?
It's been a while.
Oh, really? Oh, I was on a panel. I was on an interview panel. He's stuck in Bath like myself. So anyway, Nick does a lot of exciting things. He classifies himself as a behavioral ecologist, and he studied organisms. Some of you know it's called D-Melanogaster. You're supposed to know what D means. D-Melanogaster. Anybody knows what D-Melanogaster is? Yeah, hands up. D-Melanogaster. Is it a horse? Is it a bird? No, yeah. You know, but not everybody. Yeah, not everybody. So basically, fruit fly, tiny little things. And if those of you who are adventurous, he offered to invite Gabor. I can't see Gabor Shampoo here, but hopefully he's coming. He will take Nick to some of the planning cafes that still learns in English if you want. And just for purely scientific
Research.
It's pure research, nothing fun element at all. So medicine work. So if somebody wants to join, please let us know. So after the seminar, we go for Mensa. Please do join us. Have questions and chat with me. The department pays everybody. The department pays the lunch of everybody.
No, the department pays the guests.
Oh, sorry, not even me. Oh, sorry.
We are part of the department.
Yeah.
So pays there's this young gentleman, and after this, I think if you want to socialize a little bit, chat with Nick Banamis, he will be around until 5 p.m. They're all welcome. Well, Nick, all yours.
Yes.
Thank you.
Great. Well, thanks so much for the invitation to be here. It's wonderful to be here. I have to say, a little bit of this is wondering where Tamás goes when he goes to Hungary. And
I don't go to that still. I must say.
Okay.
I don't know what it is. So.
But it's, I have to say, even after my first morning here, it's obvious why Tamás comes. I mean, the behavioral ecology and the research happening here, let alone what a beautiful city. It's sort of an ideal place to be a scientist, really. A corner of the world where you can work. That's really what we're all looking for. And I have to confess also that I change what I say I am, depending on what the audience is. So I'm a behavioral ecologist today, but I was at an entomology conference, and I tried to say that I was an immunologist. But I have to say, my heart is really in behavioral ecology, and I'll try to explain why in this talk. I don't know should we turn off this light maybe? I don't know, or I don't know if we can keep that one on. Other way around. Yeah, other way. Maybe the other one?
No,
Oh, we can't. There we go. Okay, perfect, yeah. So, so this image here is of a pair of mating fruit flies that we captured, flash frozen, in a minus 80 degree freedom. So you had to very carefully put in male and female flies, and then as they're mating, gently move them into a tube, which then immediately went into a minus 80 degree freezer. So they're trapped in copulation forever. And then put through actually something very similar to the CAT scan that you in the hospital. And what you can see, this sort of says what's going on in the fly. You know, there's muscle up top. You can see ovaries there. Actually, I think this is the male reproductive organ. It inflates in the female that big. And this sort of encapsulates the type of research we're trying to do, is that we're trying to investigate how microbes transmitted from male to female, very low dose infections, how that then alters the reproductive decisions of that female, and what is the implication of this. Now, if you think about it, most insects don't cuddle. Maybe in some cases there's parental care, in Drosophila there's not, but they tend to stay away from each other. So the potential for for transmission, disease transmission, other than through sexual congress, is very low. And it turns out this sort of highlights a missing aspect of immunity research. And in particular, these low-dose infections, potentially more ecologically relevant infections, might be actually more what's caused the evolution of the immune system, and might explain evolutionary constraints much better than any method we're using today. Okay, and that's what we're focusing on, is how these low-dose infections impact the female, and in particular, looking at reproduction. Because it turns out how we define immunity, the innate immune system, is the set of intrinsic mechanisms designed to improve the survival of organisms post-infection. How it's defined prevents its own evolution. And in fact, reproductive responses to infection are referred to as non-immunological responses. So how it's defined can't even influence reproduction. So there's two huge blind spots to immunity research, which is low-dose infections, no one studied it, and then reproduction. It's a huge unknown. Okay. So I think I can, does that work here? But that's fine. I can also do it this way too. Okay, so behavioral ecology is where it's at. Why am I talking to you about fruit fly behavioral ecology? There must be more interesting organisms out there. And I have to say, I have tried. I really have tried to leave. That works now. Great, thanks. Yeah, perfect. And the reason why I get pulled back into the flies is because we can resolve big problems just in the fly. So how does immunity evolve? And actually, even with the fly, we can address big classic problems in behavioral ecology in ways that we can't in any other system. So for example, where are the trade-offs? We've all read in textbooks about evolutionary constraints, how organisms can't optimize every trait simultaneously, and therefore that's a major constraint. And the argument is that's why animals behave the way they do. And in fact, behavior itself is one of those things where we expect behavior to be good, right? It wouldn't have evolved unless it provided advantages. But we also expect it to be costly, because why wouldn't they show that behavior all the time? So we need to understand the context. And that means we could need a scale that allows us to work out where are the evolutionary constraints. And then this general question about terminal investment, it is fundamental to behavioral ecology theory. Going back to Williams and Hamilton and all the classic papers that many of you have probably read. But is it really terminal investment as described by the theorists? As far as we can tell, we can only find one true test of the theory that involves residual reproductive value. And what we're going to show, what I'll show you today, is actually it's probably not terminal investment. It's probably more just costly investment. And I'll explain that distinction. And then, fundamentally, why we need the fruit fly is that identifying the trade-offs and working out important aspects of classic theory and solving problems coming from fields in molecular biology, it's not enough. What we really need to do, if we're evolutionary biologists, if we're behavioral ecologists, we need to figure out the why. And the why is also a how. So underlying all of those trade-offs, there is some physical constraint, like a molecule that won't perform under a certain context, and that's what causes a cost. And so we've been investigating whether things like basic nutrition could be the major constraint in evolution. The reason why we don't live longer and have healthier guts and are able to fight infection. There is some physical reason. And unfortunately, that's why I get pulled back to the fly every time, is that just you can get a scale that you can't with many other species. Well, beautiful species. I hate being stuck in the fly lab. But, and then, Rook, okay, this is the take-home message here. Ultimately, it comes down to being able to count babies and lifespan and measure very accurately demographic characteristics. Because you can figure out how things help and hurt, and when they help and hurt. Effectively, you can work out how it's helpful, when it's helpful, under which context. And again, the fly gives us a scale that we can sort this out in. Okay, so that's a kind of long introduction. But this last point here, I just want to explain this. Sometimes, physical constraints. What do I mean by this? Okay, so I'm going to ask you all a question here. You're going to be part of a live survey. How do long-lived flies live long? And what I'm going to ask you to do is raise your hand if you think which one of these it is. Now, I should point out that this question, how do long-lived flies live long, this question has probably resulted in something like 40 Nature papers, possibly in the triple digits of Nature papers, depending on what you mean by a long-lived fly. This is sort of core to what the biology of aging and healthy aging people do. So we've evolved lines that live long. So the question is, why do they live long? Have they evolved because they live a long time because these flies eat less? Maybe they've evolved their own caloric restriction. maybe they've evolved to change their food preferences. So they only seek out diets that help them live longer. And there is a lot of research showing that actually high carbohydrate diets, at least for a fly, help them live longer. Have they evolved longer telomeres? Is their gut less permeable? So that way they have better barriers against gut dysbiosis and sepsis. Or have they altered how they process the nutrients? So they all eat the same, they just take nutrients differently. So what I want to say is this: if you solve any one of these convincingly, that's a Nature paper. And I would say that's an unsatisfactory answer. Really, this is an unsatisfactory question. This is what I mean by physical constraint. We don't need to know, okay, now we found this gene that does it. But that's useful, don't get me wrong. There's a lot of good, important research that happens that way. We need a biophysical explanation for what constrains trait expression, what constrains the trait optima. And if we can find that and then link it to the effects of those genes, now we understand. So all those Nature papers still haven't solved aging. Nobody's won the Nobel Prize for solving aging. Just want to point that out, even if they get lots of Nature papers out of it. Okay, so that's what we're looking for, is a biophysical explanation. Okay, so now we're going to do the survey. Can I get hands for how have the flies evolved eating less? Have they evolved changing their food preference? Okay, good. Have they evolved longer chromosomes? All right, you can put your hands up to multiple though. You don't have to choose one. Sorry. All right, have they evolved better gut permeability or lower permeability? Okay, that's. And what about nutrient processing? Have they evolved better nutrient processing? Okay, so, right, okay. So, so the answer to all this is option C. So it turns out that they haven't evolved eating less, they eat the same amount. And in fact, oddly, males and females choose the same diets, even if it's suboptimal for their sex. Just, they don't change their food preference. In fact, actually, this was a bit of a trick because they get fed a consistent diet. There's no choice in there. But when we do choice trials, they don't choose any differently. There's no chromosome lengthening evident in insects. It's actually a characteristic of yeast and mammals that we find telomere differences is, and the vast majority of living creatures don't show changes in telomere with age or physiological stress. There's no evidence of any changes in gut permeability. And we've done a series of experiments now where we do carefully controlled experiments measuring protein intake and carbohydrate intake. And then, this is the tricky bit, biochemical assays on the poo of those flies, of individual flies, to work out how much protein is processed and how much carbohydrate is processed. So the crazy thing here is what's happened with these long-lived lines is that they poo out incredibly high levels of unprocessed protein. So the Bradford assay, it just shoots up, skyrockets with high levels of protein. So something really weird is going on there with their digestion. And we've been able to now show that actually this causes them to have a higher infection risk. So there's a major trade-off there that living long actually causes them to be more susceptible to fungal infections, contact fungal infections. Okay, so this served the point of describing a lot of research we've done, and also this point about physiological constraint. I think we're all on board with that now. And so this is some of what we've done is very carefully set up experiments where we had individual flies in tiny tubes eating out of tinier tubes. So this is like individual flies in centrifuge tubes where we drill holes, and we have capillary tubes that have known ratios of diets. So in this case, our experiment had 1, 2, 3, 4, 5, 6 different ratios of diets. And then we diluted those diets down and then asked how long do they live and what's their reproductive output. And then we mapped onto that, then using a 3-dimensional software, we then statistically asked where are the peaks in this, and that's what the color coding is showing. Where it's high shows where the highest lifespan of a female is, for example. So you can see females in general live the longest on a very high carbohydrate diet and very little protein, really. And this X there, that's actually when we've done choice trials to work out what do they actually choose. So they do a pretty good job of choosing a diet that optimizes lifespan. But what we found in the females, this is what we reported on in our aging cell paper, was that actually they choose diets that are lousy for offspring production. So there's a trade-off there with their diet choice. And oh yes, this is what these little tubes look like. I mean, a fly is tiny, and they're eating out of these. And actually, the diets we're feeding them, we developed a holytic diet. So that's a diet that's just like chemicals off the shelf of carbohydrates and amino acids. So we can be very precise. We know exactly how much they've had. And then by collecting their poo that they leave in the tube, we can work out how much they've digested. We haven't done that for every single one of these flies, just to point out. So the take-home message here is that protein for a fly is bad, but sugar is good. And I just want to mention that when we've done this for infected flies, we've done the same grid for infected flies, the peaks totally shift. And actually, it's a peak down here is really where the optimizes lifespan and reproduction for an infected fly. And in that case, you can see here the rail is a higher protein. So overall, it's a lower dietary intake is better. But in this case, actually, protein is good and sugar is bad if the fly is infected. So infection is dramatically shifting the diets that optimize reproductive success. So, so this has sort of gotten us thinking a bit about, okay, hold on a second here. This aspect of nutrition affecting the ability to fight infection, and the point about reproduction, infection altering reproduction, neither of those are in innate immunity research. Those would be termed non-immunological, nutritional effects, and are non-immunological. And when you work it out, Drosophila was famous for finding the innate immune system, but actually potentially more infamous, and I'll even say this online, for setting up a system where you think of innate immunity as only the mechanisms found by particular labs that have found these mechanisms, not actually how animals fight infection. So these incredible pathways, I mean, they have won, there have been Nobel Prizes for discovery of innate immune pathways. This may not be how animals fight infection in nature. Even though this is conserved, we have this same pathway, this may not be how we typically fight infection. And I just want to point out this one gene down here, DIF, which is pretty central to upregulating the antimicrobial peptides, which are then thought to be a key part of mechanisms fighting, attacking microbes. Now, one of the major problems, so how have they missed this? Why am I so confident that there's a major problem with immunity? And actually, their immunologists would be aghast at me saying this, she went this out. Well, how this was done was by stabbing flies and injecting them with very high doses of bacteria. So what that does is it misses the defense of the cuticle, I mean, our skin is actually our best weapon against infection, even if it's not, you know, a fancy mechanism. And then the problem is, and then they die within hours. So it's not too surprising that nobody's studied reproduction, because there's no way for the fly to even respond fast enough to mount a physiological response that matters. And these immune genes have been found because they improve longevity for a few hours. And of course, they are specific. I mean, don't get me wrong, this is still good research. You know, so this part of the pathway attacks gram-positive bacteria and fungi, and then there's another pathway that affects gram-negative bacteria, and we've been able to show this in lots and lots of different organisms. So it's not to say this it doesn't help fight infection. It's just there's a door opening for maybe we're missing a big part of how animals fight infection. And again, this question of is it survival or reproduction? So what we did was we developed the thing you would have thought we would have done already, which is to give flies low-dose infections. What we do is instead of stabbing them with the needle with very, very high quantities of bacteria, we collected fungus that's the most high fungus in a fly if you squash it or if you even let a fly die and let's see what it grows out of it. And we just ran the needle through a lawn of that fungus from the fly. And then we very carefully tap the thorax of the fly with that fungus. So incredibly low dose infection. So this is a before picture. And then this is after we tapped it. That's what it looks like. This is on the surface of the fly, very, very low dose. And in fact, then we can quantify this, that with one tap has a very low fungal load. This is in spores per milliliter, because we give it the same washing solution to work out what's there. And if we tap them 3 times, we're getting comparably similar but more load. And this is what it is if we dust a male. So we'll drop males in a flask that has spores in the bottom, and we would just stir the male around for 11 seconds. It totally coats the male. So actually, an STI and the 3 dose have pretty similar loads. So this is actually then we allow that male to mate with a virgin female. That would then be our female with an STI treatment. And then the DTI's treatment would be taking females and then just dropping them in that flask and lightly swirling them around. So these are all contact inoculations. The DTI's one is actually typically referred to as the natural infection treatment. But although no one has ever been crazy enough to do this. What I mean crazy is now, instead of having the experiment done within 4 hours, because the flies die because of injection, they pretty much last just as long as they do if they were uninfected. So very laborious infections, which is probably why it's in the fruit fly lab. But anyway, so tapping with low-dose infections. All right, so now we can start looking at how do the animals respond to these types of infections, so low-dose infections. And we found a really interesting pattern. If we gave the fly 1 dose, what we found was that egg viability dropped. So just a single tap on the thorax, the flies respond by reducing egg viability. If we gave them 3 taps, they respond by increasing egg viability. Now, this may sound crazy, what's happening here, but actually this is core to terminal investment theory. This is the threshold in terminal investment. The idea is that when animals get doses that they can handle, they'll just say, no, we got this. We're going to go invest in survival. And as the infectious dose gets more extreme, then they invest more in reproduction. So it makes sense that we found this, even though it was a bit crazy. And also note that when we did the 0 dose versus an STI, we see a similar boost in egg viability. Other thing to point out here is this is egg viability. When we look at egg production, just the number of eggs produced, there are no differences between any of these treatments. So this is not like a traditional reproductive investment being higher in terms of total quantity of nutrients. This is actually about the character of those eggs is changing. So the eggs are less likely to hatch to adults. And I think, again, no one has looked at these very low dose contact transmissions, but it sort of suggests that possibly the egg viability itself, just the probability that a kid will make it to adulthood, might be a huge constraint or an important constraint. And so then, when we then apply this kind of approach now to an immune gene knockdown context, is now bringing in some molecular biology of the fly. This is now one of the benefits of the fly, is we can just call your buddy up, send me your line, and they send it to you, and then you can run your experiments on it. So are immunity genes for survival or fecundity? Let's talk about it. Now in the DTI scenario, where they get a large dose topical infection, if they have full expression of their immune genes, they're expressing DIF. So we focused on DIF because it's so central to antifungal pathways. We do see that its animals still suffer from those large dose infections. So they have lower fecundity and lower longevity. So lower longevity by about 45 days, which is a lot for a life of a fly, and lower fecundity by about 100 days. But if we then knock down the expression of this gene, what we see is fecundity is reduced even more and longevity is reduced even more. So in this case, we'd say, surely, yes, DIF is important for survival, but it's also important for reproduction, which before this has been missed. But then when we look in the STI context, we find something very different. Now this is, just as a reminder, we're thinking low-dose infections might be more relevant to the types of limitations animals experience in nature. And here we find that knocking down DIF reduces survival, but it actually dramatically increases reproduction. So they actually get a fecundity boost in these low-dose infection contexts. So clearly, immune gene expression affects reproduction. And it's specific to the context of the infection. Now, let's get back to that question about where are the trade-offs, and let's look at that slide again. Essentially, what we have here is a positive genetic correlation under DTIs and a negative genetic correlation under STIs. So a positive genetic correlation, we'd expect, we would say, very clearly, this is how immunity could evolve, that by turning on your immune genes, you can live longer and have higher reproductive output. But under the low-dose STI scenario, evolution is constrained. What we mean by that is the level of immune gene expression can dictate whether they live slightly longer or have greater reproductive output. But it's not like there's an obvious, this is a line that will spread rapidly. We sort of expect under these low-dose infections that there might be a range of strategies. Some organisms might be better at fighting infection than others, but maybe they might pay for that with lower reproductive output, whereas others might be highly reproductive but pay a cost for that in greater susceptibility. Okay, so then when we looked across our experiment, we could find trade-offs between egg viability, lifetime egg viability, and longevity. So I think we typically in behavioral ecology, we think about trade-offs between total reproductive output and longevity. But actually, maybe it's just as fundamental as how often do your offspring make it to adulthood. And maybe that's the constraint. So this harkens back to Charnov's invariance. And this suggests that egg viability might be one of those invariants that's constraining optimal life history. Now, one thing that's kind of interesting is we see negative relationships or trade-offs for the 1 doses and 3 doses, and also for the STIs. These were done in 2 separate experiments, is why they're separated. But with the large dose infections, we actually don't see evidence for that trade-off. So if this would suggest that actually in the context of a large dose infection, it could evolve greater ability to fight infection without incurring changes in egg viability. But note, in general, egg viability is much lower in a DTI context than an STI context. So this suggests that actually we find evidence for the trade-offs that are predicted by life history theory when we look at infections that are perhaps more ecologically relevant. And maybe this is why immunity research has missed these kinds of life history trade-offs. So, right? It's the behavioral ecology here, right? So it looks like an evolutionary constraint that's determined by infection strength. So does low-dose infection stimulate terminal investment? So, of course, what we showed was a reproductive boost. We haven't actually shown that there's any kind of change in survival, but it turns out reproductive boost is correlated with a change in life history. So that 1-dose treatment where there was a reduction in egg viability, actually that's the treatment that lives the longest. It's the yellow one here, the highest than the control. Whereas the 3 doses and STIs, those are the ones that got the reproductive boost, but they have significantly lower survival through life. And again, just want to, maybe I should be in an immunology lab finishing experiments within hours. This took us, 100 days to finish up. So there is a cost to trying to be ecologically relevant, even in a fly lab. And then, of course, the DTIs with this fungus, when females are coated with the stuff, they suffer greatly in the early period. And I think what happens is they clear their infection, and then subsequently their survival is pretty normal. So basically, yes, this low dose infection response is consistent with terminal investment. But there's a problem with the theory here. So this is what I mean about being able to take what we learn in the fly lab and apply it to classic questions. So Williams in 1966 and a whole set of classic papers in behavioral ecology since then have argued, but actually never tested, this idea that current reproductive effort and residual reproductive value are expected to have a negative covariance, which is to say, if animals show high reproductive effort at any time point, then we would expect to see lower reproductive performance later in life. And actually this is built into the theory. This is what causes terminal investment. This is what they argue causes terminal investment is that they have lower reproductive output later. But when we measure residual reproductive value, because again, if you measure the babies, if you count the babies and measure mortality rate, you can actually calculate these estimates. We have seen no evidence for the expected decline in reproductive output. In fact, the 3 dose actually had very high reproductive effort, expected reproductive output early in life, and that maintained through life. We would have expected it to be lower, especially late in life, but we see it's actually higher. And it's the same when we look at the STIs. STIs stay high. We would have expected it to be low if it was consistent with the theory. And then in the DTIs, where it didn't show a reproductive boost, it found no changes in egg viability. Actually, that's where we see drops in expected reproductive output. So basically, we find no support for this idea of that the costs are asynchronous from the benefits. So the alternative, of course, is that the benefits are simultaneous to the costs. That it's not a delayed effect through life. Animals pay the costs at the same time that they're getting the benefits. So using a whole set of age-specific model fitting, we've been able to show that that's what happens. So for example, for the 3-dose infection, we see an early age boost, and then it kind of declines. In the STIs, we see an early age boost, and then it declines. And this is saying that there are immediate benefits, and that those decline over time, as evidenced by the infection by time interaction. And we see the same thing with age-specific mortality. So the costs in survival are early, but they decline over time. So this is showing that the control has a lower mortality rate than the 3 doses and the STIs, and that is reduced over time. So rather than it being a terminal investment, it seems it's just like it's an investment where the costs are paid immediately. And we've never had a test of this theory before. And I don't think it would have been possible to do this in any species but Drosophila, just because of the sheer numbers needed to get these demographic estimates of age-specific mortality rate and reproduction. There's, I've tried to explain how we can use behavioral ecology to clean up the messes of people in molecular biology. And we can use the fly to test fundamental questions about trade-offs and terminal investment. So people have struggled finding these trade-offs that are supposed to be limiting evolution. And people have just assumed that terminal investment theory is correct. But I think with this low-dose infection system, I think we're finding ways to address these questions in a way that we really haven't been able to do before. So I just want to get back to this identifying physical constraints limiting evolution. And I had mentioned before nutrition and temperature. And I should say, this is my PhD student, Jingbo. This is a shout out to you, Jingbo. Came to me with this figure yesterday of an experiment where we were looking at how flies seek out different temperatures when they get infected. And specifically, flies normally prefer 25 degrees, and they seek out 22 degrees when they get a metarhizium infection. And we wanted to ask the question of, okay, well, what are the contexts under which this is a beneficial strategy? Can we actually show that, in general, habitation at cooler temperatures is a bad idea, except if you have an infection, and except if the nutrient conditions allow it to happen. So it's a complicated thing here, but fundamentally it comes down to behavior may not evolve except under contexts where it's beneficial. And the beauty of the behavioral variation we find in nature today might have only come about because there are specific contexts when it's really useful to have that behavior. And then it makes sense that they would only exhibit that behavior in certain contexts because it's costly. So this is the figure Jingbo gave me yesterday. And what we're looking at here is the black dots are the longevity and reproductive output values at 22 degrees, and the squares here are at 25 degrees. And essentially what we're plotting here is just for each treatment, what is that the longevity of the organism and what is its reproductive output. And so basically we can think of nutritional choice as potentially dictating a reproductive strategy. So, and in general, what we find is that, the temperature they live at is, constrained in various ways. So if they live at cooler temperatures, they tend to live longer, but they live at warmer temperatures and they have greater reproductive output. So, and in every case, it's constrained, except if they have, if they're in a high yeast condition and they have a metarhizium infection, in which case there's a serious advantage to living at cooler temperatures. So this suggests that the temperature-seeking behavior as an immune response is only adaptive under particular nutrient conditions. So there's this link here between nutrition and temperature for a fly. And fundamentally, the protein itself is then dictating whether behavior can evolve.
Nick, so what is this difference? It's really, really puzzling. So from 5 figures, there's actually a trade-off. You mean longevity and reproduction, right?
Longevity and reproduction. That's what it is. Yeah.
Not on yeast. What miraculous things happening on
Yeah.
yeast?
So basically, this was in an insane experiment.
Females, right?
Yeah, it was something like 90 for each of those treatments. Very hard to administer. And all it was we had a standard fly food, and then standard fly food where a single grain of yeast was dropped, brewer's yeast was dropped to the top of the surface. Now, in general, we've established that flies get higher reproductive output, but also we established that they have shorter lives when there's yeast, and that sort of some of that holds up as well. So you can see.
It looks like the yeast removed the constraint, right?
Yes, the yeast removed the constraint. Yeah. Yeah, and we've now. Jingbo is probably 2 days away from producing the figure to show that actually what's happening is the effect of the microbiome on the fly changes with temperature and nutrition. So if they don't have additional nutrients, and actually we've been able to work it out, a single extra amino acid, then the microbiome doesn't change sufficiently. So there's this neat link between the insect, the microbes it contains, and the ambient condition. Okay. Well, thanks so much. Yeah.
Questions and comments? Yeah, please. Yeah, go ahead. Yeah, nobody's here. Yeah, who wants to get going? Yeah, questions? Louis? Yeah, please.
Yeah, you mentioned this, probably I was that when you apply this DSTI treatment, then you don't have a trade-off. You just have an increase in both reproduction and longevity. And in the STI, you have this trade-off. And what is the reason for this? Because as I understand that the DTI.
Yeah, that's a direct topical infection where they get coated in the fungus. Yeah.
That's a harder infection or
Yep.
more dangerous. And they still be able to invest in both reproduction and longevity. In another treatment, that STI, you have the stable. So why is this? Why cannot the fly invest in both in the smile, under the smile?
Yeah, so if you don't mind me going back. Two things. One is, I mean, this effect of diff on longevity is established, and it's true even for stabbing. They live longer when they're stabbed and injected with lots of bacteria. They live longer, and in some ways, it's not surprising that in our context, it takes 4 days for the fungus to get through the cuticle, to get in the hemolymph. So it's not too surprising that we can measure reproduction, and that reproduction, they're more likely to reproduce. Because if the fungus gets in quickly, it would kill them quickly. So this aspect I don't think is that surprising. I think it is a bit more surprising that we find this trade-off. And we don't necessarily have an answer to that except when you put the two of these together. Let's remember, this is the same gene. What it's saying is that diff isn't an immunity gene. The immunity genes themselves are the expression of the immunity gene is dictating the life history strategy. And it's doing it in a strain-specific way. Now, what I actually think is happening is that we've been able to show that when they get large dose DTIs, large dose infections, the animals, excuse my language, shit out whole protein. They become incredibly protein deprived. Internally, they become sugar rich. And then that creates conditions that allow for gut dysbiosis and then sepsis, which then is probably what kills them. For a large dose infection, we find no evidence of microbes in the hemolymph. So for like an STI here, it looks like they're actually able to fight the infection, but it's enough. But they have to pay enough of a cost to clear those infections, and maybe that's why we're seeing this trade-off. And in fact, what we find is when they get STIs, they improve their absorption of protein. So a fly normally processes about 85%. When we give it amino acids we can then estimate that about 85% of that is consumed because we don't see it as unprocessed protein. And that increases to 90% when they get an STI. So they actually have an internal gut management. And then this is one of the cool bits I was telling you about. We're trying to work on self-medication. Didn't talk about that today. But we've now established that alcohol that the flies seek from the natural yeast, that improves it another 5%. So they get up to something like 95% ability to absorb protein if they have an STI and they're able to find natural chemicals in the environment. So I think this is about protein management.
Thank you. Yeah. More questions? Yeah, go ahead.
Sort of a two-part question, actually. Firstly, why did you pick this specific strain of fungi to run these experiments with? Why not bacteria? And second, do you think these trends would hold true for a bacterial rather than fungal?
Yes. So thanks for the question. So there's a study by Weberly which showed that it's something like 85% of all known potentially higher of all known infections in Drosophila come from STIs, and they've only ever identified fungi that are transmitted. So that's not to say bacteria don't have a role, but in terms of the types of transmissions that happen between insects. That being said, I think it's a fair point, because it could well be that what they eat has bacteria in it and they need to clear it, and actually the gut itself is another epithelial layer similar to, so it's not exclusively to fungi. I mean, I also got to be honest that we use fungi because they're easier. I mean, although we have seen shifts in nutrition alter fungal load within the fly, and we did a lot of work to work out what's relevant and which ones alter. But you can grow up spores very easily. The spores are, if they're mature, they become fine powder. We don't actually have to break them up with chemical compounds, and that means we can then apply them just dry to a fly. Bacteria are typically much more difficult to administer. One of the bacteria STI paper takes individual flies and dips them in bacteria, and then estimates transmission from a male to female. So it's just a more ecologically relevant system. It relates to the highest infection found in the fly, and it's easier. Yeah, and that's probably the most important answer. Yeah.
So Nick, great talk. I'm just wondering about this terminal investment logic. So if I understand you correctly, the logic is that if you give an infection to the beast, then somehow evolution programs this beast to allocate the resources in a strategic way. But of course, the alternative explanation is simply the organisms are struggling. So it's simply there's not a strategic decision, but his or her body has to fight with some diseases, and the way it shuns the energy to fight with diseases, this is how you get survival and or reproduction. How do you separate these two?
Very difficult to separate. It's an excellent question, and I think particularly because we were able to show that the costs show up at the same time as the benefits, we really can't exclude the possibility that terminal investment is just they're struggling, and they're trying to achieve something. And then of course you might think, is it an optimal allocation for both? But that's and that might be able to get at that question, but yeah. So it's a conundrum. I mean because these infections have nutritional demands, I actually think a very compelling hypothesis is that because there are protein demands, by increasing protein levels, the flies are then almost forced to reproduce more. Because protein is available, the biochemical process for reproduction is there, and maybe there's no adaptive consequence at all to any of this. And actually, maybe it comes back to the more fundamental question, which is, what is the probability that the egg will make it to adulthood? And in every single one of these experiments, the treatment affects egg viability. And maybe egg viability is actually what is the critical thing, and we've been missing a trick with thinking about number of eggs. It's offspring quality rather than quantity.
Last chance. Yeah. Okay. One in. Yeah.
Thank you for the presentation. I don't know if it was about flies, but it raised the question for me: is do these infections can have similar or the same effects on the viability of the sperm cells?
Oh, great question. Yeah.
How do
So,
these affect?
yeah.
Who cares about males?
Yeah. No, so we have not addressed that question. But you know what? Actually, getting back to the easier answer, sperm are actually easier to sort out because we can. There are simple staining techniques for us to establish whether sperm are viable or not. I mean, so that's actually a question. I take that one home because we really should have done this already, and particularly as it relates to egg viability.
The reason why I'm asking is your very first image, the frozen copulating flies, and you said that small dose infections can occur when they mate. And that could also be, for example, if the male isn't really urged to its terminal investment, then so it could be because of the effect of the infected.
Yeah. So what we've been able to establish is that during mating, females get twice as much load from a male than a male would get from mating with an infected female. And, and we have found behavioral changes in infected males. They copulate more rapidly. And actually males are much more likely to transmit infections to subsequent females, and females hardly ever pass it on. So I think that sex specificity about viability actually is a very relevant one and could be core to what causes sexual conflict known to occur in flies. So that's a really good point, and it's testable. That's the, yeah. Yeah. Great.
Great question. Thank you very much for Nick for the presentation. And remember, in a few minutes we just hang around, so do join us for this fancy Mensa food, which is 5-star, 25 Michelin star chefs. So do join us, okay?
Yeah. Thank you, everybody. Yeah. Do I get? Are you